So now I would like to introduce Andrea Luppi. Dr. Andrea Luppi is a research fellow at the University of Oxford, Cambridge, and McGill. And his work focuses on computational models of altered states of consciousness and the structure-function relationship across species. So please, Andrea.
All right. Well, thank you all for being here. So let's get started. Well, I started off my journey not as a neuroscientist, but as a philosopher, and I've been interested in this question of what it is that's going on through our minds. And that, in a sense, is an ever-changing landscape. It's like trying to map an atlas of the clouds. And in fact, over the course of a few seconds, you could go from being engaged with the previous talk to being bored with the current one, to hopefully being engaged with the rest of this talk all over again very quickly. And yet, even though all of these mental states correspond to different brain states, the structure itself of the brain over that short time scale remains pretty much the same. So how is this possible? Well, in fact, it is the same kind of question that we all have to deal with when we're navigating a trafficky road. For example, the Riot of London. We might want to go from one place to another, and we know that depending on the time at which we're trying to navigate those same roads, the traffic is going to be high or low, and we might need to change the path that we take in order to get to destination in time. So I'm going to propose that we take this traffic metaphor seriously for now, and we think, what happens when we are in a start state and we're trying to reach a target state? Well, some target states are easier to reach than others. For example, if you're going downhill, that's easy. Going uphill, not so easy. And that means also that transitions differ from one place to the other, depending on which direction you're going. And in the case of the brain, of course, we don't have a map like this one, but rather the map that we can think of is the human connectome. The map, the wiring diagram that connects every region to every other region. And in turn, we then need to ask, how can we formalize this question? In fact, it is a question that engineers have been dealing with and physicists for a long time. And it's called the question of natural control. Now, as I said, I'm a philosopher by training, not a mathematician. So although this is the proper way to think of this problem, it's not how I like to think of this problem. And I prefer to think of it as a few ingredients that we're putting together in the right order to answer a question that we have. So we have a target state and a start state, and all of those will need to be defined further later on. Then, as I mentioned, we have our map, the wiring between those target states and start states. And we need a control strategy. We need to know how we're going to approach moving into this landscape. And finally, all of those previous ingredients put together will tell us how much effort we're going to need to go from where we are to where we want to be. And if we reorganize all of this a little bit differently, then we find that the first four ingredients are the determinants of the fifth. And we can ask a more interesting question, which is, when is effort low or high? Well, effort is going to be low when the target is close to the start. It's also going to be low if there's a convenient path linking the start and the target. For example, if you've ever been to the UK, lovely place I also recommend visiting, you might find out that two places like Oxford and Cambridge, despite being geographically close, are actually quite difficult to navigate because you have to go all the way through London and then back. And any direct bus will take about 5 hours. That may or may not be from personal experience. So the location on the network matters. Is there a convenient path? In that case, the answer is sadly no. And finally, you can choose a smarter or less smart strategy. And if your strategy is smart, and you're applying your effort where it can give you the best outcome, then that effort is also have to be lower, everything else being equal. So how do we apply this to study transitions between states, which is, after all, the topic of this symposium? Well, for example, we can take them one piece at a time. So let's start with the target and the start state. Now, you might wonder, what is a target state? What's a start state in the brain? One option is to define any of these states in a random matter, random activation across the entire brain. And just for context, when I mean a brain state, I mean a pattern of activation across every region in the brain. And I'm mostly gonna be talking about cortex here, so apologies for the more subcortically inclined. Now, one option, as I've mentioned, just random patterns. You can generate as many of them as you want. The problem is it's not obvious what meaning they would have or why you might want to go from this random pattern to that random pattern. So another option is to use fMRI tasks. Then we're going to know exactly what that pattern means. It's the pattern that occurs when people are undergoing a particular task. But studying transitions between those is going to be very difficult because you're going to need, if you're interested in many different states, then you're going to need all of the possible combinations of them, and that takes a lot of time to acquire. So instead, we're going to take one of those smart shortcuts, or so we hope, and use Neurosynth instead, which is a meta-analytic engine that summarizes a lot, thousands of research studies into brain maps associated with different mental contents, or so the story goes at least. And so if we apply now this trophic metaphor seriously to the brain, we can define start states and end states as different Neurosynth brain activation maps. And we can use this network control framework to ask how much energy, how much effort does it take to go from one of these to the other across the human connectome. So we're going to keep the layout of the roads fixed, that's the connectome, and we're going to try to move from one Neurosynth map to another. For example, going from the attention map to the language map. And if we do that, which we can, and all of this is purely in silico, then we find that there is one striking pattern and comes out of here. Namely, although it is often said that it's not about the destination, it's about the journey, when it comes to transitions in the brain, it's not about the journey, it really is about the destination. Some destinations, as you can see here, are just that much harder to reach than others, pretty much regardless of where you're starting. And although I don't have time to delve into that in this particular talk, I might like to point out that some particularly human-specific brain maps, like language, seem to be among the hardest to reach. And maybe that is why there's only so few species that have language. Now, although we did use Neurosynth because of its convenience, we also wanted to make sure that we had a more biologically realistic validation. And in fact, we can replicate all of these results when we look at actual transition between tasks in the Human Connectome Project actual task fMRI data. And we find once again that language is by far the most difficult pattern to reach. And we also find that going from a 0-back task to a 2-back task, so a more difficult version of the same working memory task, is harder than going from the hard to the easy. Going from hard to easy is easy. Going from easy to hard is hard. That's what the model says. Makes sense. We were very happy to see that. So we've looked at the target state and the start state. How about we look at the network now, which until this point we've been keeping fixed as the human structural connectome reconstructed from diffusion MRI. How much energy does it take on average for all of those transitions? So now we're averaging across all of those 123,000 transitions. It turns out that the human connectome is pretty good because, for example, if you're trying to navigate between Neurosynth maps, not on the human connectome, but on a randomized version of the human connectome, that's much, much harder. It's a much less favorable layout of roads. Now, of course, if you're just randomizing the connectome, that's not going to fit inside your skull. And humans have only limited cranial capacity, so it's important to preserve some of these more biologically meaningful properties of our networks because human brain networks are actually spatially embedded. And if we try to preserve that spatial embeddedness in our random graph, two things happen. The first thing is that layout becomes a lot better for favoring transitions between Neurosynth maps. So that's the good news. But the interesting news, I'm not sure if it's good or bad, is that the human connectome is still better than the geometry-preserving randomized null. In other words, we figured out part of the magic sauce that makes the human connectome so good as a layout for favoring transitions, but we haven't figured out the full story yet. There's something beyond geometry in the connectome, in the topology of the connectome, that explains why the connectome is so good at facilitating cognitively meaningful transitions. And last but not least, we want to look at the control strategy. How are we favoring those transitions? Up to this point, we've been implicitly assuming that you're inputting your effort equally into every region to steer the transition to the next state. But you don't have to do that. You can actually change the local distribution of where you're putting more effort. For example, if you happen to have a disorder, maybe your brain is thinner or thicker in certain regions. And if we, certainly an oversimplification, but if we simulate a transition in a brain that has perhaps less capacity for exerting effort in some regions, because those regions are thinner compared to what they would be in a healthy brain, then we can figure out what kind of transitions will become easier or harder than the healthy brain. And it turns out that it's not a uniform phenomenon. You can't predict what every transition is going to be just looking at the pattern of cortical thinning and thickening. You have to look at the transitions themselves. Conversely, many of us will know that medication is a useful way of changing the state that one is currently in. That's one of our main avenues of clinical treatment. And ultimately, one of the ways that medication works is by engaging different neurotransmitter systems. And those neurotransmitter receptors are not distributed equally across the cortex. Rather, thanks to the work of Dr. Justine Hansen and others, we now know that many different neurotransmitter receptors are distributed heterogeneously across the cortex, and we have maps for that shown here. So if you simulate engaging different neurotransmitter systems according to where there is more or less of those, and you simulate that as an increase in your control energy applied to that region, once again you find that some neurotransmitter systems seem particularly promising for facilitating some transitions. And vice versa, there are some particular states where it might be easier or harder to reach, and some of them are not particularly facilitated by any of the receptor maps that we're considering here. Again, this is by far an oversimplification, but hopefully it's not a useless, but rather a useful simplification that we hope can guide further modeling efforts and ideally clinical translation. But all of this has been taking the traffic metaphor seriously. But how seriously can we really take that metaphor? Because we know from different cognitive neuroscience, but also imaging and theoretical perspectives, that in the brain, not everything can be happening at once. There is, for example, some might say, a global workspace where different stimuli are competing for our attention, and some of them need to prioritize over others, meaning that there is a competition in the brain. And we can see that at the macro scale as well, with different resting state networks being anti-correlated with each other, meaning that when one is more active, the other is less. So we're starting to have this idea of competitive systems in the brain, which we do not get in our traffic metaphor. All roads in that sense are equal. So how can we study this? Well, you've seen already that I'm a fan of modeling. And so in this case, we can take empirical functional MRI data and empirical diffusion tractography data and try to integrate them into one model. And we use a Hopf model, which is a simple model of oscillators that can show both oscillatory and non-oscillatory dynamics. And then we update iteratively every connection in that structural connectome that we started with until our model best replicates the actual activity and connectivity that we see in the real data. In other words, we're trying to approximate the effective connectivity of the model. Now, none of this is new. This is already well established. There's been papers for at least 2 years, if not more, about this. The only change that we make is that we also allow competitive interactions. In other words, we allow the model to change the sign of the edges in the connectome from positive to negative if doing so will produce a better fit. We don't force the model to do it, we allow it to. And when I say competitive, I don't necessarily mean long-range inhibition. I am being agnostic about the underlying biology. Whatever that might be, the outcome of that is in our model. If region A is more active and it's connected in a negative weight to region B, then region B will be less active. This is all I'm saying, being agnostic about biology. I want to be clear about that. So the question then is, what does that give us? Do we actually get anything from that competitive interactions? And because we know the diffusion tractography has limitations, we apply this model not just in human, but also in macaque and mouse, where we have track tracing data that are fortunately not typically acquired in humans. Now, if you use the standard Hopf cooperative only model, you can do pretty well already. 0.42 correlation is very well compared to a lot of the literature. But we can do more than twice as better if we allow those competitive interactions. So that they're giving us a lot of extra predictive power, and not just at the group level, but also at the single subject level. And not just in the human. We can do better in the macaque, which was already doing quite a bit better than the human because we have better tractography, both group level and subject level. And even in the mouse, we're doing even better and better. So those competitive interactions are buying us something in terms of explanatory power. And the models are not just generically better at looking like a human brain. Your own model is going to be better than my own model at predicting your brain activity. So the models with the competitive interactions are more subject-specific and better discriminate between people, which is useful because if you want to make a model of a patient, you want your model to be a good model of that patient, not of any other patient. And just for the more computationally inclined, if we start asking whether this model is capable of recovering any form of ground truth, the answer seems like it's yes, because if you simulate from a known structural connectivity, from a known, effective connectivity, and then ask the model to recover that, you can recover the original ground truth connectivity with more than 0.8 correlation. So it's very good at recovering the ground truth when that ground truth is known. And you're not just getting better at simulating the functional connectivity, which after all is what the model is optimizing. Rather, you're also getting better at simulating other properties that the model was not optimized for. For example, in all three species, you're getting more realistic dynamics in the form of better matching the empirical metastability. We're not quite at the level of the empirical metastability yet, but we're much better than the previous model with only cooperative interactions. And if you take that connectivity that you've obtained from this model, that effective connectivity, and you use it as a reservoir computer, so you use it as the wiring diagram of an artificial neural network to perform a working memory task, all of a sudden, allowing competitive interactions improves the model's ability to perform that computational task. Again, just one simple task, but it's giving us something that we did not optimize for. And finally, we don't even need to look at the whole activity and connectivity. We can look at individual patterns of co-activation that happen at individual time points and ask how well do they match the kinds of patterns that we would see from neurosynth. Do they look like real brain patterns, or do they look like random collections of regions activating that happen to activate together? And in the real human data, indeed, there's a high correlation between individual patterns of activity at rest and the neurosynth patterns that we all know and love and that I was talking about before. That is not the case for the cooperative-only model, but adding only those few little competitive interactions all of a sudden gives you a lot better ability to replicate those neurosynth patterns in your simulated resting state data. And finally, because computational modeling is so powerful, we can combine or go towards combining the two previous approaches, the one enriched with local properties and the one that simulates the data. So we can have more biologically realistic models than the one I was just talking about and perform all sorts of perturbations on those. We can perturb the global wiring, we can lesion individual regions, and we can enrich regional heterogeneity based on disorder maps or based on neuromodulatory maps, as we have seen before. And then we can evaluate those same sets of perturbations in terms of thousands of readout functional properties, not just the few that I was talking about before, but actually a lot more. And this is just a snapshot of some of the functional outcome measures that we can look at, and you can look at all of 6,000 and odd of them because we have made all of this available on our website, so you can read the preprint, find the link, and then play around with the data at your will. So altogether, we hope that we can convince you that modeling can really provide a Rosetta Stone to link symptoms and cognition to underlying network organization and its dysfunction, and then the local micro and chemoarchitecture that we observe in the brain. Now, I'm already out of time, but I just want to thank all the people that have made this work possible, because it takes a lot of people for this kind of work. I did not collect any of the data myself, so to all of them, my deepest, sincerest thanks for being part of this journey. And if you want to have a look at the fuller set of results and at the full story, then please engage with these publications, preprints, and you can also find me online. Thank you all.